Okay, ladies and gentlemen, we're now moving on to our next student presentation. Erik Estigoy will take us through Mortal Kombat, the technological advancements behind the fight against aging. Take it away, Erik.
All right, so hey guys, my name is Erik, and like Dr. Saul said, I will talk about what technological advancements have been made to fight aging. I don't know if you noticed, but the title is actually a pun on a video game with the same name, Mortal Kombat. So that's where I got the title from. So it seems like humans have always been obsessed with the idea of the ability to live forever. You can see this in Greek mythology with Tithonus. In Harry Potter, there's Nicholas Flamel. There's Claire Bennett in the show Heroes, and there's the Cullen family in the movie Twilight. And all of them have different ways to achieve immortality. In the Greek myth, Tithonus got his immortality from Zeus. In the book, Harry Potter, Nicholas Flamel got his immortality from the Philosopher's Stone to make the elixir of life. Claire Bennett had the innate ability to regenerate herself, and the Cullen family, well, basically, they turned into vampires to gain immortality. So all these examples are from work of fiction. So is there an immortal organism that's living on Earth right now? Well, you don't need to look further than in our oceans to find out. There's this jellyfish called Turritopsis dohrnii, or the immortal jellyfish. So why is it immortal, you ask? Well, here's a picture of a normal life cycle of a jellyfish. So it turns from a fertilized egg, it goes through a larva, and then to a polyp, and it turns to a medusa, which is the jellyfish itself, right? Well, the Turritopsis dohrnii has a unique spin on this life cycle. The medusa will live a normal life where it'll turn to an adult medusa, and after it mates and reproduce, it can turn back into polyp. So why does it do that? Well, during times of stress, like there's no food or there's environmental hazards, it can revert back into a polyp, or basically transforming its cells into a mature cell to a youth cell, or going back to youth. So scientists and researchers have wondered if immortality is achievable in humans. Would it be like this jellyfish right here? So according to the British Institute of Posthuman Studies, we have technology and resources right now to combat aging today. So to investigate if we do have the technology and resources, first we must define aging. The American Heritage Dictionary defines it as the process of growing old or maturing. This definition doesn't really give us any way to see if technology can fight it, or technology can explain how to fight it. So I'm going to use Aubrey de Grey's definition of aging. He's the guy with the awesome beard. He's actually a researcher and scientist. His works such as mitochondrial free radical theory of aging and ending aging. He also has been in a bunch of TED Talks, so all those works have made him one of the leading people in the anti-aging movement and made super longevity in the current world right now. So his definition of aging is the accumulation of all damage in a molecular, cellular, or whole body, in the whole body. He has 7 ways. He says he has 7 ways to accumulate aging. There's cell loss, cell atrophy, senescent or toxic cells, nuclear mutation, epimutation, mitochondrial mutation, extracellular crosslinks, extracellular aggregates, and lysosomal aggregates. Now, for each age-related damage, he also has a way to repair them. So for the purpose of this presentation, I'm just going to talk about cell loss and cell atrophy and how the technology can fight aging via regenerative medicine. So what is regenerative medicine? Aubrey de Grey defines it as any intervention that seeks to restore the structure of a tissue or organ to its state before it suffered damage. So there's a lot of ways to have regenerative medicine. One way I will talk about that is the most newsworthy and encouraging is stem cell-based therapy. So stem cells. What are stem cells? Stem cells is a single cell that can replicate itself to become another stem cell, or differentiate itself to many cell types. There's two kinds of stem cells in the human body, the adult stem cells and the human embryonic stem cell. Adult stem cells come from deep within the body, and they include blood stem cells, cardiac stem cells, and neural stem cells. These stem cells are classified as multipotent in that they can become one of several related cells, but not any other cells. So for example, blood cells can become lymphocytes and platelets, but they can't become neural cells like interneurons or glial cells. So even if they're already specialized, adult stem cells are actually being used in the medical field right now for various reasons. For example, hematopoietic stem cells or blood stem cells are being used for bone marrow transplants to treat bone marrow disease and blood diseases. And there's also been other cases like this one, where a blood stem cell transplant can cure a person with HIV in it. So while adult stem cells can serve multiple purposes for treatment and research, they're only limited to their ability of only creating cells that are related to them. So what we're interested in are pluripotent stem cells, which is a human embryonic stem cell. These are pluripotent, like I said. They come from the inner cell mass of a blastocyst. So pluripotent stem cells are cells that can differentiate into any cell in the body. Since human embryonic stem cells are undifferentiated, they cannot be used directly to treat for therapy, but they can be differentiated to a specialized cell where it can be used for therapy. So these pluripotent cells can be made from two sources, via in vitro fertilization or somatic cell nuclear transplant, or SCNT. So in vitro fertilization, the first person who made human embryonic stem cell was James Thompson. What he did was he made in vitro fertilization with a sperm and an egg, fertilized it, took the morula, turned it into a blastocyst, and then took the inner cell mass from the blastocyst and plated it in an embryonic stem cell media, and that's how he got the pluripotent stem cells. So certain problems arose with his cell lines. Like for example, it was very expensive to make them. Immune rejections, since human embryonic stem cells are different from our own bodies, so the chances of the stem cells creating more problems is there. And ethical concerns, since the blastula had a potential to become a person. Question the viability of using these cells for research and treatment. So other researchers tried to answer these problems. One of them was Samuel Wood. What he did was creating stem cells via somatic cell nuclear transplant. What that is what he did was he removed the egg from a nucleus, so it's an empty nucleus, and then he took another cell from the different body, like let's say a fibroblast, so a diploid cell, and he fused that diploid cell with a nucleus to have fertilized egg, and then he did the same thing after, where he let it grow to a blastocyst, and then he removed the inner cell mass to have a pluripotent stem cell. Now, this removed the ethical reasons because it was not made from an egg and a sperm, so there was no potential of being another person. But there were still two problems in the way. So other researchers, ethical concerns are removed. So other researchers have tried to solve the next big problem, which was immune rejections. So multiple studies have been found that cells that are already specialized can be induced to become pluripotent stem cells. So this is Shinya Yamanaka. He's actually one of the founder of induced pluripotent stem cells, or iPS cells. He actually won the Nobel Prize for Medicine in 2012 for this discovery. So what they did was, first they needed to identify which genes were highly expressed in embryonic stem cells, right? After the researchers used viruses to introduce overactive versions of these genes, they found that the differentiated cells could turn to a pluripotent cell by having only four genes, which was KLF4, SOX2, CMYC, and OCT34. They then compared their iPS cells with embryonic stem cells, which is the ES cell right there, the Entera2, which is a cancer cell, and HDF, which is a human dermal fibroblast, where the cell came from. And as you can see, the iPS cells made the same proteins as the embryonic stem cells, and not the same as a human dermal fibroblast. All right, so since the cells can be created from the same person who needs them, immune rejection is no longer a problem. So how can iPS cells then be used for aging? So to combat aging, Rando and Chang explains that iPS cells can be used to fight aging by reactivating the enzyme telomerase, which is responsible for maintaining telomere length and the long-term self-renewal of embryonic stem cells. By reactivating telomerase, the life extension can be possible since there's multiple studies that have shown that telomeres may be responsible for aging. However, it is known that telomerase is also made by cancer cells, so it can provide unwanted risk if telomeres are extended. However, there was a study by Lapacette et al. In 2011 where they used iPS cells from old individuals to rejuvenate old cells and without providing side effects of cancer. What they did was they used iPS cells from old individuals, and then they undifferentiated, right? And then they redifferentiated to another fibroblast. So it's basically the same thing, but they wanted to compare if it's actually younger or not. And as you can see, the iPS cells have the same effect as as a human embryonic stem cell compared to a fibroblast, which is the old senescent cell. And another proof is the heat map here, where the differentiated cell has different gene expressions compared to the old fibroblast cells. So this is when a skin cell is de-differentiated and then turns to another skin cell. I'll show you another study where they actually turned a skin cell to a different type of cell. This was made by Yamanaka and all again in the same lab, where they used human pluripotent stem cells to become either cardiac cells or neurocells using different biochemical cues. Here you can see it actually turned to a neural cell. The D is differentiated and it has different gene expression compared to the undifferentiated, or the N-terra, which is the cancer cell, or HDF, which is the human dermal fibroblast. And then the next thing they did was they actually differentiated to a cardiac cell right here, and then it actually beats. So there's actually video evidence that they actually made the human fibroblast to a cardiac cell, which I think is pretty cool. Yeah, cool. So age-related diseases and disabilities, such as Alzheimer's, could be treatable by using these cells. Since the stem cells are made from the person in need, immune rejection is not a factor anymore. And unlike stem cell or organ transplantations, patients do not have to wait if they are genetically similar for transplantation to happen. In addition to replacing dying or dysfunctional cells, the ability of engineering new cells and tissues can be used to study disease mechanisms and drug testing. So the use of induced human pluripotent cells sidesteps the ethical reasons as well as the immune rejections. Still, clinical use of this technique is actually not ready. Yamanaka's lab has shown that the use of a virus to insert the transcription factors actually made cancer cells, so there's a risk to that. There's also another study where they said it was very inefficient to make iPS cells, where it was only 0.01% to 0.1% actually turned to a pluripotent cell. It was very time consuming in that it took 2 weeks to actually make the pluripotent cell, and then the vast majority of the cells failed to remain pluripotent. However, there's current research right now in the iPS cells that include finding a safer and cheaper way to generate iPS cells. So one way is putting the 4 genes in a transposon, which is a mobile gene. How it happens is have the 4 genes need to induce pluripotency in a cell, be located in transposon, and then it temporarily inserts the 4 genes into one of the cell's chromosomes. The transposon can then be removed by the host transposase after the iPS cells has been created. So the study here, the efficiency of inducing iPS cells could actually increase up to 20%, and the risk of cancer formation is lowered since you're not using a virus anymore. It could also be induced using stimulus trigger, such as if you put a cell in mild acidic condition, it will actually turn pluripotent. So again, it will reduce cancer risk since you don't need to put a virus in that gene anymore, and you just need to put a cell in mild acidic conditions to make a pluripotent cell. So while the tissues derived from these methods are not ready for transplantation for therapy yet, the results that have been found are an important milestone for research in fighting aging and may soon be enough for clinical and therapeutic use. So what if there was a way to redifferentiate cells without going back into stem cells? So remember the Thriptopsis dorni, or the immortal jellyfish, where it can revert from an adult medusa back to a polyp repeatedly. Well, the way that the immortal jellyfish rejuvenates itself is with a process called transdifferentiation. Transdifferentiation is a unique biological process where a differentiated cell can become another differentiated cell. So for example, a skin cell can turn to a motor neuron cell. So how can we use this knowledge to aid the progress in the fight against aging? So here's an example of a way to do it. As discussed before, cells that are differentiated can be induced to become pluripotent. So a fibroblast cell can become induced pluripotent cell and then turn to a neuron after and then transplant it. Transdifferentiation offers a different way to do that, in that they have a fibroblast culture and then use transcription factors that know that is produced by neurons, and just go to a fibroblast to neurons without having that middle step right there. And then it becomes the neurons, and then you can purify and transplant it again. So since transdifferentiation does not require the middle step, it can actually provide a faster way to provide healthy cells or tissues. So with this idea, any specialized cell can become any other cell by receiving the right signals. So is this actually achievable in humans? Well, there was a study by Aida et al. In 2010, where they stated that they were able to reprogram mice fibroblasts into functional cardiomyocyte-like cells that contracted without creating iPS cells. So what they did was, in vivo, they created the induced fibroblast cells with 3 developmental transcriptional factors, GATA4, MAF2C, and TBCX5. Those 3 were actually sufficient enough to reprogram the fibroblast cell into cardiomyocytes. And to test if these induced cardiomyocytes behave like a normal cardiomyocyte, they implanted these cells into the hearts of mice. And the results they received were pretty astonishing, in that they found the induced cardiomyocytes actually behave like normal cardiomyocytes after implantation, as you can see with the action potentials and the beating of the heart. So what are the implications of this study? They said that the avoidance of the dedifferentiation of a fibroblast cell to a pluripotent cell before cardiac redifferentiation would actually greatly reduce the cancer cell formations in stem cell-based therapies. In addition, the fibroblasts can be grown using cardiac or skin surgery in vitro for transduction of defined factors to transdifferentiate the cells, and can be followed by the delivery of the cells into damaged tissues. Also, in the experiment, the injection of the fibroblast cells into the heart after 1 day of inducing the 3 transcription factors resulted in the reprogramming of transplanted cells. So what that means is the result implies that there can be a potential to introduce defined transcription factors or factors that mimic those effects directly into the heart to reprogram fibroblast cells that are present in the heart already. So future considerations for the technology against aging. The quest for immortality is actually getting closer due to recent technological advances and research findings. However, the results that were produced from the research are from laboratory work, so a lot of things are controlled, and it doesn't really portray outside world instances. We also still do not know a lot about the human genome. New discoveries are being made every day. There's actually like a theory, or not a theory, but studies that said that human genome is not the way, but gut bacteria is the way to cure some of these diseases, since the gut bacteria in our body actually help a lot in maintaining our health. There are also other ways that super longevity can be achieved other than stem cell-based therapies, like epigenetic drugs, elimination of damaged cells, telomerase reactivation, and clearance of senescent cells. So there can be other ways to actually find it. It's just stem cell-based therapy is the most newsworthy right now, and it's actually the most promising. So the results gained from the studies are promising. The new technologies are being made faster and more efficient at an exponential rate, which can increase the speed of research in the field of aging. So super longevity may be seen in the foreseeable future, and individuals today may see the dawn of a new age. Yeah. Any questions?
So this is an area that I myself have realized that I need some direct education in it, and so I'm going to a regenerative medicine conference May 5th through the 7th. So future students will be learning from me a bit more directly than you guys, where I'm intensely interested in this, but I don't feel I have the direct knowledge that I really should have, and it's a very complicated area. The other thing that I want to tell you, you may have noticed me sending an email during Erik's presentation. Why would I do that? Well, Mallory Chipman, who performed at Future Day, was in this CBC Searchlight Canada-wide competition. And she's just won the first round, and she's now into the second round. So it's a very exciting thing. It's very hard to get there. Most people stop at that first round. And so you can all help. You can vote for her every day now, between now and sometimes some weeks from now. Anyway, I just want the third thing to tell you is some sad news about time. That we really, I'm very sorry, but we have to move on to the next person, or I will be in deep trouble at home.